Hi everyone, welcome to the next session in the microbiology high yielding points. The first two sessions, I hope you've seen the same. The first session was on the special stains that we use in microbiology. The second was on all the microscopes and the questions related to that. Today we are going to cover another part of general microbiology, and that is bacterial genetics. So under genetics, the question that usually comes in the NEET PG exam and the topic that I've taken up under the high yielding point is how genetic material gets transferred. So first, I want all of you to know that there are 4 methods that I'm going to tell you in this particular transfer of the genetic material. And yes, let's begin without any further delay. I think you know I'm going to talk about transduction and 2 types of transduction. I'll talk about conjugation, I'll talk about transformation, and once in 10 to 12 minutes I've finished with all of those, we are going to go and attempt MCQs related to that. Also, thank you for the amazing response to the previous videos. I've been reading all the comments under the same for the microbiology crash course, and I'm going through each and every answer that you guys have been giving on the previous videos or the homework that I'm giving you. So yes, I think I'm going to make that a routine to give you a little homework, because that obviously makes you search for that homework in that video, and again it's training your brain to answer something. So yes, thanks a lot for answering, and I hope it helps you in the exam too. So let's begin, guys. This is bacterial genetics, and the first thing, so listen to me for 10 to 15 minutes, and then we have lots of MCQs, all previous year questions. When we talk about how genes get transferred, the first method that I want to tell you is transduction. Now transduction is of 2 types. We can have generalized transduction and specialized transduction. I'll go one by one. Please remember, for transduction, the most important thing that you need is a bacteriophage. So how does a bacteriophage look like? I hope you remember the bacteriophage has a classical tadpole-like morphology. It has a classical tadpole-shaped structure. And actually, what is a bacteriophage? Is it a bacteria, a virus, a parasite? So remember, bacteriophage happens actually to be a DNA virus. So a DNA virus is going to help in transfer of genes between bacteria. So let me show you with the help of an example. The first thing that I'm teaching you is generalized transduction, and over here comes the bacteriophage. It has gotten attached. So there are 2 cells, okay? There will be one bacteria which is going to be the donor bacteria, and then in the end there's going to be one bacteria which is going to be the recipient bacteria. So currently I'm working on the donor. It has to donate the chromosome, the DNA. How is it going to donate? Who's helping it donate? Bacteriophage. So what does bacteriophage do? Bacteriophage comes inside, attaches over here, and the entire bacteriophage doesn't enter. It sends its DNA. So you can see the DNA has entered. So now do you agree you have 2 types of DNA in this cell? One is the DNA that the bacteriophage has gone. One is the DNA which the bacteria anyway had. So now when new bacteriophages will form, some of them will have their own DNA, some of them will have the bacteria's DNA. So the ones which have their own DNA, they are the usual ones. But the ones which have got this red color, the bacteria's DNA, they can now go and donate that to the next bacteria. So can you see the red color bacterial DNA has now been donated to the recipient bacteria. And now has the donor and the recipient bacteria managed to recombine? Has recombinant DNA formed? So what do you understand by generalized transduction in simple words? It is carried out with the help of a bacteriophage. Bacteriophage comes inside, picks up the donor DNA, gives it to the recipient DNA. This is a general process. Let's put a little bit of twist in the story, and now let's call it specialized transduction. So what do you mean by specialized? It's the same bacteriophage, it has given the same DNA, but this time it's not before new things are formed. Both these DNAs decide to enter into a pact. That is, the DNA of the donor bacteria, the DNA of the bacteriophage. This time they're not separate. They decide to first make a pact. They first recombine with each other, and after making a pact, they plan to separate out. So have you seen what happens next? First they decided to, first they've recombined, and then they end up breaking up. So when they detach, when they again come back, they tend to form a hybrid. Can you see, this is just not the bacteriophage DNA, it's a combo. The part that has detached is a little bit of both. A little bit of both has detached. So can I say the final part that we have is a combo of the bacteriophage and the donor DNA? Now from these, new bacteriophages will form. So can you see, new bacteriophages that have formed, they don't have, if you compare it with the previous one, in the previous one, when the new bacteriophages were formed, either they had their own DNA or they had the donor's DNA. Now over here, the bacteriophage that is being formed, they have a combo, they have the recombined DNA in all of them. And now can we say all these recombined DNA, they are going to go and give this recombined DNA to a recipient. They are going to go and give this recombined DNA to a recipient bacteria. So in a way, once again, has the donor bacteria, the donor DNA reached the recipient DNA? Yes, but this time, did it go only as a single thing? No, this time it went in recombination with the bacteriophage DNA as well. So it's pretty much the concept is the same. If you ask me what is happening in generalized transduction versus specialized transduction, in both of them the bacteriophage is being used. In both of them the concept is that the donor DNA has to reach the recipient DNA. However, the only difference is that in generalized transduction, the bacteriophage is only going to carry the donor DNA with itself. Whereas in specialized transduction, the bacteriophage is going to carry actually a combination of the donor and self DNA. That's the only difference. Having said that, we also have to study sometimes what can happen in this process. There is something known as lysogenic conversion. What is this? This is a very unique thing other than what I've taught you. What is lysogenic conversion? The story is still the same. Can you see? You have the same bacteriophage. The same bacteriophage has given its DNA. So this is the bacteriophage DNA. This is the donor cell DNA. And both of them, can you see, they've again entered into a pact. But are they forming new bacteriophages, or are they detaching and then all of that I taught you? No. You say that once it's fused, it remains the same, and the cell undergoes division, just like mitosis happens. Cell undergoes division into two daughter cells. So remember, the parent bacteria had only the parent DNA. But now do you see the two daughter bacteria, they end up having the combo DNA of the parent and the bacteriophage. So will they have some new properties? Other than the parent DNA, little bit of bacteriophage DNA has also come into them. So will they start displaying new properties? Yes, and that is known as conversion. They are getting converted from their parent. They are having some new properties, so they are getting converted. Hence, I call it lysogenic conversion. This is an examiner's favorite previous year question. We'll also be practicing a few. Guys, with that, transduction is over. Generalized, specialized, and a variant, that is lysogenic conversion. Now I move on to the next one, and that is conjugation. So very simple. Again, over here, this is your donor bacteria, this is a recipient bacteria, and now the transfer of genetic material will occur. But am I using bacteriophages? The first step, that is no. Bacteriophage is not used. So then what is used for the transfer? I make a tunnel, I make a connection, and that is known as, that is going to help in the transfer of the genetic material. So what is this bridge or this tunnel known as? This bridge or tunnel is known as the conjugation tube. So yes, you can call it the conjugation tube, you can call it the conjugation bridge. I'll write it for you. Either you can call it the conjugation bridge, or you can call it the conjugation tube, or you can also call it the pili. These are actually nothing but pili. I'm going to talk about this more. But first thing that you have to know is, do you agree that the donor will have its DNA? Yes. But apart from DNA, is there something else which the donor also has? What is that something else? That something else is known as the plasmid or the fertility factor. Repeating, the donor has its own DNA, which is understood, right? The recipient will also have its own DNA, that is understood. But the donor, I'm calling it a donor because it has something extra, and that is known as F plasmid. What does F stand for? The fertility factor. It has something known as F plasmid or also known as sex plasmid. So F plasmid, sex plasmid, F standing for fertility. So imagine if this has the fertility factor, I call it F plus. This does not have the fertility factor, I call it F minus. In other words, the one which has the fertility factor is going to give this fertility factor to the one who doesn't. So can I say one becomes a donor and one becomes a recipient? That fertility factor is there in one, so that's the donor. It's going to go in the other, replicate and go in the other, that becomes the recipient. So the donor is also said to be the male bacteria and the recipient is said to be a female bacteria. That is how the terminology of their genders is done. So what now happens if F plus, now we're going to do many you can say combinations, permutation combinations I'm going to give you. So if I say that, okay, can you see this, what do you want to call this? This has the F plasmid, F plus fertility factor present. So can I call it the donor DNA or the male or the donor bacteria or the male bacteria? This doesn't have the fertility factor, so I call it the female bacteria or I call it the recipient bacteria. Now what am I going to do? I am going to replicate. We are going to replicate this plasmid and give it to the recipient bacteria. Via what? Via that same bridge. No bacteriophages over here. So this is going to replicate and going to the recipient. So now have both of them become F plus F plus? Many students feel that this is going from here to here, so the donor will now become F minus. No. It replicates and a copy of it goes into the recipient. So this will continue to have the F plasmid that it had. This will just get a replicated copy of the same. So can I indirectly say that when F plus and F minus they conjugate, both of them end up becoming F plus F plus. So we are going to do some reactions. This is reaction number 1. When F plus and F minus, repeating, when F plus and F minus conjugate, they are both going to end up being F plus F plus. Let's consider scenario number 2. Scenario number 2, before I get into this, again I want to ask you, this is a donor bacteria. Do you agree it's going to have its own circular DNA? Like any bacteria, it will have the DNA. And like I'm telling you, the extra chromosomal part, that is going to be the F plasmid, the fertility factor. So do you want to call it an F plus cell because it has the fertility factor? Before this fertility factor goes into someone else, they end up having a pact. So they end up getting fused. The donor's DNA and the F plasmid end up getting fused. What do you call that cell? Recombinant cell, right? Both of them recombined. So we call it HFR. High frequency recombinant. Remember, we call it high frequency recombinant. That is what you have to know. Having said that, now let's consider next permutation combination. If I have a high frequency recombinant cell, this is a high frequency recombinant cell, with the help of a conjugation tube, if that is conjugating with F negative, what is F negative? Does it have a plasmid? No. Does it have fertility factor? No. So what will happen when they conjugate? Let's see. So this is HFR. Do you remember what was HFR? HFR was the fusion of the plasmid and the DNA. So what happens from this HFR? A little bit of both, means a little bit of the plasmid, a little bit of the donor DNA is going to replicate and go into the recipient. I'm saying little bit of the fertility factor is going, little bit of everything is going. So little bit of fertility factor, a little bit of donor DNA goes. So tell me, is this going to actually become F plus because a little bit of fertility factor was going? No, it is still going to remain F minus. It will still remain F minus. Entire fertility factor has not come. But on the other hand, a little bit of the donor DNA has definitely come. A little bit of the donor DNA has definitely come. So hasn't it become like too many DNAs? A little bit of F plasmid has come. A little bit of donor DNA has come. It has its own recipient, its own DNA on already it has. So can I say somewhere a fusion can occur? They all can recombine. They all DNAs can recombine, but at the same time, even though they will recombine, will it ever be a F plus? No, because at the end of the day, the fertility factor that came was little bit incomplete. So although it will be changing into recombinant, but it will remain F negative. So I'm repeating for all of you. If we take it up in a very simple manner, once again we'll see that when there is fusion of HFR with F negative, a part of plasmid, a part of DNA goes into the recipient. All those part things go and combine with this recipient's DNA. So the recipient's DNA definitely becomes recombinant. But since a part and part is going, the fertility factor is not completely gone, so it still remains F negative. So what is your formula now? That when a high frequency recombinant and F negative cell conjugates, high frequency recombinant cell remains the same. That F negative cell also somewhere remains the same, only recombinant F negative is formed. Ultimately, I realized everything ended up remaining the same. HFR remains HFR, F negative remains F negative, only that it's got a recombinant DNA. Let's go on to the third and the last one, that is known as sexduction. What is that? So let me take a test. What is this? This is the donor DNA. If this is the donor DNA, you'll say this is the normal donor DNA. And what is this round structure? This is the plasmid. So this has the fertility factor. Is it F plus? Yes. If both of them end up having a pact, if both of them end up integrating, what will you call it? You will call it high frequency recombinant. You've studied that. However, after entering into the pact, if they break up and now this plasmid says, I'm going back into my original state. But can it break up so easily? Won't it carry some memories? After a breakup, memories always reside. So while separating, the plasmid took a little bit of the donor DNA also. So can you see over here? This is not a pure red color plasmid. It's got a little bit of green color, a little bit of memories from the donor DNA have come. This plasmid is now, it cannot be called as a regular plasmid. It is known as F prime. So first and foremost, till the plasmid is separate, original F plus. If it fuses with the donor DNA, recombines, it is a recombinant HFR. If it again disintegrates and comes back with some memories, you call it F prime. Guys, when F prime conjugates with F negative, now can you see this is the recipient F negative, it doesn't have a plasmid. When F prime replicates and goes into F negative, this also becomes F prime. So can I say that when F prime and F negative recombine, both of them end up becoming F prime, F prime. Let us do a quick revision. Tell me, when F plus, a regular simple F plus, was recombining with F negative, what were both of them becoming? Both of them were ending up becoming F plus, F plus. If F prime is going to conjugate with F prime, if F prime conjugates with F minus, both of them end up becoming F prime, F prime. However, if recombinant conjugates with F minus, they pretty much remain the same. They remain the same recombinant, they remain the same F negative. Only thing is this F negative also becomes a recombinant DNA. So easiest way of learning, if simple things are written, in the end everything is going to become that. If you're dealing with F plus, in the end both of them will become F plus. If you're dealing with F prime, then both of them will become F prime in the end. Only when you're dealing with this HFR, they tend to remain somewhat the same with this recombinant form that is present. Now, having said this, the last type of genetic transfer is the easiest. Just pick it up and eat it. As simple as that. So, I've taught you transduction, that needed bacteriophages. Second, I taught you conjugation, that needed plasmids and the conjugation tube. Third, I'm teaching you transformation, which is very simple. So, guys, this is a bacteria. And it says, am I going to use a bacteriophage? No. Am I going to use any conjugation tube? No. I'm going to directly pick up DNA fragments from the environment. So probably there was a donor DNA. There was a donor DNA. And you can say this was a donor bacteria. This was a donor bacteria. That donor bacteria has somewhere gotten lysed. So you can see the donor bacteria is somewhere getting lysed. Once the donor bacteria gets lysed, what will come out of it? The donor DNA is going to come out of it, and that DNA will simply be taken up by the recipient bacteria. Simple. So what is transformation? If you just have to learn in 2 words, transformation is uptake of DNA. That is it. It was actually first discovered on Streptococcus pneumoniae by Griffith, which is a previous year question. This transformation was discovered, and all of this finishes off your genetic transfer. Let's practice a few questions, then I have another point to tell you. Okay, so question number 1, lysogenic conversion is, so is it the integration of the host bacterial nucleic acid to the phage, integration of phage nucleic acid to host, bacterial mechanism to cause antigenic shift, or bacterial method to acquire resistance? I hope you remember lysogenic conversion. It was like the daughter bacteria were getting some new properties. Why? Because the phage nucleus and the host genome were getting fused. If you can't recall, then remember this was I'll quickly take you back to what was lysogenic conversion. This was lysogenic conversion. What was happening in this? You could see that the phage DNA over here and the bacterial DNA, they were getting fused. They were getting fused together. So that was resulting in lysogenic conversion, phage into host. Coming to question number 2. What is the mechanism of direct transfer of free DNA? What do I mean by direct transfer? Is there any bacteriophage involved? No. Is there going to be any tube involved? Yes. Is there any tube involved? Direct transfer. Direct transfer of free DNA. Direct transfer of free DNA. So think again. This is one of the most, this is actually your homework for the day. So I know it's going to create a lot of confusion. Is there any tube involved? Is there any bacteriophage involved? So that is your homework for the day. This is a very famous PYQ which has been asked 3 times in the exam earlier. And I want all of you to tell me direct transfer of free DNA. Will you call it transduction, transformation, or conjugation? 1 hour after this video is uploaded, I'm going to give you the final answer. So I'll be looking out for all the comments after that. Okay, moving on to question number 3. What is a transduction? So first and foremost, think in your mind, what was a transduction? Was it using a bacteriophage? Yes. Was it using a tube? No, it was using a bacteriophage. What is a bacteriophage? Bacteriophage is nothing but a DNA. It is nothing but a DNA virus. So now let's start reading further. Is it bacterial mediated viral recombination? Viral mediated? Yes, viral mediated. Viral recombination? No. Viral mediated? Yes. Bacterial recombination? Correct. It's mediated by viruses, but I'm dealing with bacterial recombination and transfer. Moving on to the next question. Can you identify this particular GIF? So it moves slow, but you'll understand. There's a death of a donor bacteria. All the DNA has gone out. Donor DNA fragments have ruptured. This is a recipient bacteria. And this recipient bacteria is just going to take up those DNA fragments. Take up the DNA fragments and recombine them. Did you use any bacteriophage over here? Any bacteriophage used? No. Has there any conjugation tube being used? No. Have a look at the GIF again. This is the donor bacteria. There's first going to be death of donor bacteria. The DNA, soluble DNA fragments are released. Then there is a recipient bacteria. This recipient bacteria is just going to directly take up that donor fragment and combine it with itself. So I hope you could answer. This is a classical case of transformation. Coming to the next question. This is the GIF and the homework that I gave you yesterday. What is this GIF showing you? Where a proper between the donor and the recipient, see how the fertility factor or the plasmid is going. Can you see there's a proper tube that is formed? Have a look at it again. You have number 1. You have a donor, you have a recipient. There in the center, there's a tube that is forming and this fertility factor has gone. So this was a classical case of conjugation. Let's move on to the next question. What is the end result of F factor mediated conjugation? F factor. So write in your mind. F factor means the cell is F plus and the other one by default must be F negative. Tell me, when F plus and F negative conjugate, do you agree both of them end up becoming F plus, F plus? So can I say both the strains become F plus? Correct. But I still want to read everything else. Transfer of entire bacterial chromosome occurs. Is the entire chromosome occurring or the plasmid transfer is occurring? Plasmid or the fertility factor is occurring. It converts the recipient to F plus That is right. F minus say F plus. Donor to F negative. No. That is what I'm trying to teach you again and again, that when there is a conjugation that occurs, this plasmid that you're talking about, it replicates and a copy of it is going to go. See, a copy of it is going to go. This remains as it is. If this was F plus, this will always remain F plus. So it's not going to change into F minus, not at all. So the first statement was your answer over here. Both strains will become F plus. Moving on to the next question. The discovery of transformation came from the study of one of the bacteria. I just taught you transformation was given by Griffith, and it was studied first on which bacteria? It was studied on Streptococcus pneumoniae. Next question. A bacterial cell in which F plasmid is integrated with the chromosome. Let me draw this. For example, this is a bacterial cell. This is the F plasmid, and they are saying this is the chromosome or the DNA. Now they are saying that the F plasmid and the DNA have integrated, they have combined. What do you want to call it? We would want to call it high frequency recombinant. As simple as that. Plasmid with chromosome, HFR. Moving on to the next question. Conjugation does not involve which of the following? Bacteriophages, HFR, F factor, plasmid. I hope you remember, conjugation does not involve bacteriophages. Bacteriophages have a role to play in transduction. Bacteriophages have a role to play in transduction. Moving on to the next. Which of the following term describes the relationship between a virus and a host where no new viral properties are produced and the viral genome is replicated with the host genome? So asking you again, which of the following term describes the relationship between, there's a virus, by all default they are talking probably about a bacteriophage, and they are talking about a host which is going to receive it, where no new viral particles are produced, means more bacteriophages will not be produced. Only thing is that the genome of the virus, the viral genome and the host genome will get integrated and go on to the daughter cells. I hope you could remember this was lysogenic. Lysogenic transformation. I told you it's an all-time favorite. Again repeating what they had told you over here. They said that there is a virus. The viral genome and the host genome integrate. They integrate. And there are no new virus productions that occur, only they integrate. That was exactly what was given to you in the question. It is a case of lysogenic conversion. So option number A. Moving on to the next and the last question. Again a question on lysogeny, which I know is very, very important. What is lysogeny? It's a bacteriophage that transfers bacterial DNA. Let's read further. This is kind of incomplete statement, I feel. Let's read further. A bacterium takes up DNA from the environment. This is definitely not lysogeny. What is this? When the bacteria just picks up DNA from the environment, just picking up DNA is known as transformation. This is known as transformation. Okay, coming to the next. A bacteriophage genome gets integrated with the bacterial chromosome. Bacteriophage genome gets integrated with the bacterial chromosome. Sounds like a true statement, but I'll still read the last one. Bacteriophage disintegrates from the bacterial chromosome? No. So definitely I'm certain that option C is what is going to happen in a lysogenic conversion. So guys, with that you finished transduction, generalized transduction, specialized transduction, lysogenic conversion, conjugation as well as transformation. Now there was a homework that I gave you, and that was a simple MCQ that you all had to attempt and you had to give me the answer. So this was the MCQ that you have to give me the answer to, and I am waiting for this. Mechanism of the direct transfer of free DNA. What is it known as? Very famous previous year question, and I know you are bright kids because you've been giving amazing answers in the previous sessions. So I'm not at all going to be surprised when you give me a right one here. Thanks a lot for joining in. Tomorrow, guys, we come to sterilization, disinfection, PYQs. That's the topic for tomorrow. And this is what you have under the Plus subscription. For the Plus subscription students, the good news is that you've got a 25K new question bank with clinical and one-liner questions that have been added. And apart from that, the Plus subscribers get access to the new NEET PG course, 1 month, and I think half a month left probably, but certainly a very high-yielding course for all of you. Having said that, amongst the free classes classes, you have free grand tests to give every Saturday, and then you have free special classes which are conducted at every day from my end. They are conducted at 7:45 a.m. as this kickstart morning session, and from the other educators, you know, the platform is always full of live classes all throughout the day. So we start every morning with the 7:45. I think we do a start to the live lectures in the day, and that's the kickstart morning. So that is something that I'm planning every day for the NEET PG students, and then as the exam comes for FMG students. So that's an ongoing series. We've completed approximately 153 sessions. So I think it's just never ending till your exam is conducted. See you guys tomorrow in this series. At the same time, I hope you're also following the 10 p.m. image of the day, or IOTD. So now next, I hope now we've reached the polyps and the discussion in that. So I hope you're following the same. Thanks a lot for joining in, guys. Have a great day, and see you at 10 p.m.